welcome to, I don't know, the 40th year, maybe more than 40 years we've been having this teaching series. I don't want to steal too much of Ray's time, but this started that many years ago when I was still somewhat young, just as a bunch of guys sitting around a table telling anecdotes, basically. There was almost no science involved. That's the way medicine maybe was 50 years ago. So anyway, now it's come to the point that you're going to see from Dr. Panettieri's slides how hardcore science this man does. So listen up as I previewed these slides, and I know that he's doing fantastic work in his laboratory and very kind to come out here and spend the day with us. So we'll have his presentation and then a case for him to be a visiting fireman and critique to follow. So as many of you as possible want to stay for 2 hours, you're more than welcome. So you can see from the introduction slide, current research from the Panettieri Lab. When I first knew him, he was at Penn, but now he's some years ago moved to Rutgers. It tells me that his lab is twice the size it was before. And as I said, doing brilliant work. So Ray, thank you very much.
Great.
Okay.
Pleasure. Thank you. Well, 40 years of a conference, that's pretty impressive. My only assumption, Lynn, is that you started that senior year of high school. So I want to thank everybody for coming. The other thing that's remarkable about allergy here at UW is the fact that you have a 7 a.m. conference. I know of no other allergy program that has their scientific meeting at 7 a.m. So however you can get people out, that is magnificent. It's always fun to come out because I've known what's been going on here in allergy for some time, and it's really wonderful. First and foremost, I want to thank AZ for sponsoring this trip. I do believe industry academic partnerships are the way we're going to help patients. You do your stuff very well. We do target identification, and together we enhance education and health in individuals. These are my disclosures. Should anybody know any other company I can work with, let me know. None of these is going to impact on this talk, I can assure you. Now, caution, 7 a.m., have another cup of coffee. We're going to do cell signaling. And cell signaling is really quite exciting in our laboratory. And we really enjoy doing it because we think it's fundamental to finding new targets that we can impact. We're going to identify the cell signaling pathways that enhance contractile agonist-induced sensitivity and can serve as therapeutic targets. Describe the molecular pathways by which airway smooth muscle becomes insensitive to bronchodilators. Clarify how airway smooth muscle actually plays a role, orchestrates and perpetrates airway inflammation. That's pretty strange, right? That's a structural cell. What the heck is it doing? Secreting chemokines and cytokines and modulate immunocyte trafficking and function. And then we're going to talk about an asthma airway phenotype and a new biomarker that we're exploring. We're very excited about that. New conceptual framework. If there's a stimulus in our bodies, there are pro-activating and inhibiting pathways constantly. It doesn't matter what we're talking about. That sort of yin and yang, that homeostasis will determine a phenotype. Contractile pathways can induce enhanced airway smooth muscle function as measured by hyperresponsiveness, but relaxation pathways can be just as important. And if you inhibit these, you get more contraction. So keep that in mind. That's the paradigm shift when we talk about airway hyperresponsiveness. Not many of us think about the loss of bronchodilator effect contributing to this pathway. So airway smooth muscle has actually has a job description that's substantial. It contracts, it relaxes, it grows in cell number, in cell size. It can migrate. Actually, it's one of the large cell that produces type 5 collagen and fibronectin. You can talk about myofibroblasts and transition, but essentially it is a key factor in airway remodeling. And then it plays an immunomodulatory role. We're not going to spend a lot of time talking about cell adhesion molecules by airway smooth muscle or its cytokines, chemokines. We're going to spend a lot of time up in the left-hand corner there. So airway smooth muscle, excitation, contraction, coupling. Think of this as the currency that makes a muscle contract or relax. That starts at the plasma lemma with a signal that has to get transduced to the actin and myosin, the business ends that causes shortening of the muscle. So one of the first challenges our lab had, and this goes back many, many years ago, is the airway smooth muscle fundamentally different in asthma versus non-asthma? That's controversial. Lynn probably went through many, many quad AI and ATSs where people were talking about, no, it isn't, yes, it is, no, it isn't. So we availed ourselves of a unique biophysical property, and this is magnetic twisting cytometry. So what we do is we take individual airway smooth muscle derived from fatal asthma and non-asthma, and then we take a ferrous bead, magnetize it, and then put it in a magnet and twist it, okay? And that twisting of the bead correlates with the tension in an individual cell. And we use that as a surrogate measure for airway smooth muscle contraction. Because when we grow our cells and they're on a plate, the cells can't contract, right? Because they're stuck to the plate. The plate doesn't, get deformed. So we have to measure something on the plasma lemma. So here, what I want to show you is what happens when you give a T2 cytokine, IL-13. We also use IL-13 and IL-4, but also TH1 type cytokines, or TNF, has the same function. Look what happens here. We give methacholine, we get a contraction or surrogate contraction. If in the presence of IL-13, though, the muscle's hyperresponsive to methacholine, not only to methacholine, but to any contractile agonist. So if we now take a look at asthma cells versus non-asthma cells, these are derived again from fatal asthma compared to non-asthma. Each one of these are an individual cell, populations of cells, and here you can see the force that we generate and the geographic force that's generated in an individual cell. Suffice it to say, if you come down here, whether they're early passage or late passage, if you grow them on a soft matrix or a stiff matrix, asthma-derived airway smooth muscle always contracts more. No doubt about it. The muscle, and got to remember, we take this out of the human, the donor, and this phenotype's manifested for as long as we study it, 3-4 years. So the asthma is intrinsically hyper-responsive. And that sets up a whole new story as to a more contractile cell. So here we compare asthma and non-asthma using that same kind of paradigm, methacholine and histamine. And in the asthma cells, that's shown here. In all instances, looking now at the group mean data, you can see in all this instance, regardless of the stimulus, carbachol or methacholine or histamine, the airway smooth muscle contracts more. So these evidence, and unfortunately the reference is cut off there, we published some time ago, but it sustains the concept that the muscle is fundamentally different. Well, you can do neat things with individual cells, right? So about 15 years ago now, it's surprising till it goes that fast. What we devised in collaboration with other colleagues across the country and the world is using human precision cut lung slices. So let me walk you through this. We get human lungs sent to us. These are aborted lung transplants. They could either be fatal asthma, they could be COPD, IPF. We've gotten into doing a little bit of IPF, or non-disease state. These are people who sustain catastrophic brain and heart injury, otherwise have no chronic illness. So we get lungs in a box. We take the lungs and we inflate it with low melting point agar. Underneath the plastic shroud there is ice. Low melting point agar gives the lung turgor, means that we could then cut it. And up here in the right-hand corner, we cut a slice of the lung that is infiltrated with low melting point agar. And it's a beautiful slice of ahi tuna. Okay, we get away with that early in the morning. Then we use a molecular tool, and we're going to go way back to Lynn's training. This is a cork borer that we had laying around, and we use a cork borer. This is how you used to make corks, and you put things through corks like tubes. We get a chunk of lung. We then put it on a bologna slicer. Now, this bologna slicer costs about $12,000, so it's a pretty expensive bologna slicer. And we slice 350 micron sections. And that is what you see here in this disc. And we bring it up to room temperature, the agar melts away. And what you're looking at is a human small airway, non-cartilaginous, that we can keep alive for 2 weeks. The cilia beat. The muscle contracts and relaxes. Now, why is this important? Well, I showed you the cells, right? This is now looking at all the players in the lung in situ. Now, what we don't have is trafficking immunocytes, so there's no cells, no EOs coming in here. There are immunocytes that are embedded in the submucosa, dendritic cells, ILC2s, and so forth, and we're characterizing that. But this model now allows us to contract and relax the tissue accordingly. And this is the data that we can generate. So here you're looking at a methacholine challenge, albeit in a human slice. Okay, so nobody's inhaling methacholine here. As we increase our dose of methacholine, you see we get beautiful luminal diameter narrowing, right? Constriction. This is a visual wheeze. Now we give isoproterenol, or we could give formoterol, or we could do any beta agonist. Look at this, we can relax it back to normal. We can do this repetitively 3, 4, 5 days. So it gives us a great model for target identification and studying excitation contraction coupling. So remember, I showed you that IL-13 in the individual cell, and that made it a little more responsive than the no IL-13 or diluent alone. So now we take a human lung slice and we add IL-13 at a physiologically relevant dose. And sure enough, what you can see is the carbachol curve moves to the left and upright, airway hyperresponsiveness. So in that tissue where you got all the players, epithelial cells, mast cells, the whole variety, we get beautiful contraction. So we've done some recent studies. This is, we're going to be showing some of this at the upcoming Quad AI as well as ATS meetings. We asked the question, what about IL-33? Does IL-33, an alarmin, have the ability to generate airway hyperresponsiveness? Answer, absolutely. Here's the control. Increasing doses of IL-33 moves the curve to the left and upright. So it generates airway hyperresponsiveness. And you can see here, this is the EC50, right? So if it takes less carbachol, that's hyperresponsiveness. So you can see a beautiful demonstration that IL-33 can generate airway hyperresponsiveness.
Is this true in normal tissue and asthmatic?
Absolutely. Absolutely. Yep.
Same magnitude?
No, the asthma is a little greater than what we see in the non-asthma, but its capability is in both healthy and fatal asthma. So we also wanted to look at TGF beta. Now, everyone knows of TGF beta as being critically important in matrix deposition, in scar formation, but very few people realize that it can actually change excitation contraction coupling. And here you're looking at basal tone. If I give an overnight dose of TGF beta, what we find is the airway luminal diameter goes up as compared to diluent alone over that period of time. So TGF beta can actually affect bronchomotor tone, and no matrix is being deposited, no enhanced cells. It's all about sort of basal tone. So then if we give carbachol, what you can see is in this dose response curve, the curve is shifted to the left. And although it maxes out here, it doesn't give you the same kind of IL-13 signal. It has a profound effect in the EC25 as well as in the EC50. Takeaway, TGF beta can change excitation contraction coupling that has nothing to do with airway remodeling. Why is that important? Well, think about every injury an asthmatic sustains liberates TGF-beta as a repair mechanism. That means the airway gets twitchy. And if Len and I and T are taking care of an asthmatic post-virus, they start to wheeze and they wheeze on for some time and then they repair. But these data would suggest I don't need airway remodeling to generate airway hyperresponsiveness. TGF-beta by itself can do that. So here we're looking at TGF-beta's ability at 4 and 18 hours in different doses to increase the basal stiffness. This is that B thing. We now move back to the cells. A little easier to do these in rather than the lung slices. We then use a TGF-beta receptor antagonist. Has no effect on basal tone, but it also diminishes it. So these data suggest that yes, it's through the TGF-beta 1 and 2 receptor, which that compound, the SB compound, blocks. Now the agonist-induced, I don't know how to change that little thing down there, but basically if I now give histamine or carbachol, I get an enhanced contractile response, and then I can block some of that enhanced signal by using the TGF-beta inhibitor. Now, what's interesting is when I give TGF-beta by itself, it has no effect. It doesn't mobilize calcium, and we really don't get any effect over and above carbachol if I give TGF-beta at the same time as I give a contractile agonist like carbachol or histamine. So this means it requires some kind of genomic or post-translational modification of a pathway that requires probably gene expression. Over here, we're going to go into this a little more because the way muscle contracts is it activates either GQ through a pathway that increases calcium. It's very similar to any immunocyte that's migrating, by the way, or it's getting activated. That's through release of cytosolic calcium, activates the myosin light chain kinase, and then you get contraction shown here. What people may not realize is that there's a parallel pathway in which the muscarinic receptor is dually coupled to another GQ. This is 1213, and this pathway actually activates Rho kinase inhibits the phosphatase that turns the contraction off, procontractile. But very few people realize this pathway is in parallel to this other pathway. That becomes really important in this story I'll be telling shortly. So TGF-beta can induce airway smooth muscle shortening, and it can do it through SMAD phosphorylation. I didn't show you that data, but the reference is hidden behind all this stuff down here. And it actually activates Rho kinase, and that actually is the way it can actually enhance airway hyperresponsiveness. So let's now move on to relaxation. Remember what I said, contraction and relaxation and the homeostasis. If I lose relaxation, what do I get? More hyperresponsiveness, more contractile responses. So how does that work? Well, just to reiterate, there's calcium independent and calcium dependent pathways that lead to contraction. This is very similar. This is the canonical IP3 pathway that increases calcium, and that then causes, in my cells, contraction. In other people's favorite cell causes migration. At the same time, I can get Rho kinase that inhibits the phosphatase, keeping a phosphate on the light chain, and enhances contraction. Now, the Rho kinase pathway is mediated by Rho A and through a different G protein that we've defined as G1213. So how does a beta-2 adrenergic receptor work? Well, everyone knows it increases cyclic AMP. That's not a surprise. So does everybody know what happens after cyclic AMP goes up? Protein kinase A is activated. And how? That's why beta agonists work. Beta agonists can affect the G proteins, the Rho, the sarcoplasmic reticular release of calcium, and it can actually inhibit niacin light chain kinase. So it's the perfect beta agonist, right? It's the perfect relaxant. So why are we even talking about thinking of new bronchodilators? Well, it doesn't work as well as it can. Here we're looking at asthma, non-asthma. Remember, the asthma was hyperresponsive in contraction. But when we start to look at relaxation, what we find is that the airway smooth muscle derived from asthma does not relax as well as normal tissue. Fundamentally, it's hyperresponsive, and it has less ability to relax. And that is shown here also if we use isoproterenol as a beta agonist, but that also happens with formoterol. You can see that, yes, it does track pretty well with non-asthma, but then later in the milliseconds that we're watching this, it starts to lose its ability to relax. So that's a double-edged sword, right, for asthmatics. It's not only hyperresponsive, but it's hyporesponsive to beta agonists. Well, this is a problem for our patients who go to the ER, beta-2 adrenergic receptor tolerance or desensitization. So if I take our slices and I expose them to 3 hours, 6 hours, or 12 hours to albuterol and then bronchodilate with isoproterenol, but it could be formoterol, doesn't really matter. Look what happens at 12 hours of exposure to albuterol. I lose more than 50% of the ability to relax the airway smooth muscle. That is classic beta-2 adrenergic tolerance. What happens to the patient who goes to the ER? How much of their albuterol inhaler did they use in the last day? The entire inhaler. 120 puffs. They're here. And what's the first drug we give the patient when they come in? A neb treatment of albuterol, right? That's a problem. Here you can see the Emax, it's dramatically significant. So how do we break this? What do we do? This is studies that'll lend itself to the back end of my talk here. Well, if you give steroids for 4 hours before I do the desensitization, here's the desensitization to albuterol, 12 hours. But if I then add dexamethasone or fluticasone at 100 nanomolar dose, plus the albuterol, look at this. I can't desensitize the beta receptor. Impossible in humans. You might say, well, how does that work, Panettieri? Well, you know what's interesting? In humans, there is no glucocorticoid response element in the beta-2 adrenergic gene. So it doesn't occur the same way it does in rodents, where GREs are there and they can actually drive beta-2 adrenergic receptor expression. So we don't know precisely how this works. But here is the pharmacology. I'm sort of a molecular pharmacologist. And so what we love to see is dose response and all that stuff. Here's the albuterol, but if I get dex or I get fluticasone, look at that. I cannot get desensitization. And here, giving dex actually by itself can augment isoproterenol responsiveness at baseline. I desensitize, bring it right back to normal. Yeah.
We're taught that the theoretical time of onset of action of steroids is 6 to 12 hours. You're saying in 4 hours, it's already fixed.
Oh, forget about it. At the end of my talk, we're going to talk about the non-genomic effects of steroids that occurs in people. So yes, trans activation and transrepression by glucocorticoids requires time, 4, 6, 8, 12, 24 hours. But there's a non-genomic effect of steroids where I can amplify bronchodilation to an incredible degree by giving both of these simultaneously. That doesn't require gene expression at all. So stay tuned, I'm getting there. So what if I bypass the whole system? Forget about the beta-2 adrenergic receptor and activate adenyl cyclase. That is the enzyme that generates cyclic AMP, doesn't require a receptor. If I give desensitizing dose of albuterol and then bronchodilate these are lung slices with forskolin activating adenyl cyclase, there's no desensitization at all. Why is that important? That means everything downstream from the beta-2 adrenergic receptor is unaffected by desensitizing the beta-2 adrenergic receptor. That means this isn't phosphodiesterases, nothing like that. This is all about the beta-2 adrenergic receptor. So then he could say, well, Panettieri, tell me about cell receptor number. It's got to be higher. It's got to be higher. Well, this is work we did with Steve Liggett at USF. Here's control, DEX, no effect. We're now looking at the density of the beta-2 adrenergic receptors on the individual airway smooth muscle cells. Albuterol, you lose almost 3/4 of the receptors on the cell surface. Remember, DEX repaired the whole process? Not statistically different. It is not enhancing the number of receptors on the cell surface. It's doing something below that receptor that restores the sensitivity of the beta-2 adrenergic receptors. So stay tuned. We're working on how that works. Well, what about IL-13? I showed you IL-13, one of my second slides showed airway hyperresponsiveness, right? Guess what happens when I give IL-13 and look at bronchodilation? It's attenuated dramatically. So remember the homeostasis, the teeter-totter that I talked about. In this case, IL-13 not only generates airway hyperresponsiveness, but it makes beta agonists not work as well. So you can desensitize the beta receptor. Cytokines, specifically IL-33, I'll show you in a minute, but also IL-13, actually impedes the ability to bronchodilate. So now albuterol doesn't sound like the best bronchodilator in the world. Why do we not think of it that way? Because we got nothing else to use. So when you only got one, you know, if you buy a Ford, it's got to be black, right? That's what Henry Ford said. So if you have no comparator, this is the best thing next to sliced bread. It ain't. There's circumstances where these things don't work. Here's IL-33. IL-33 profoundly in a dose-dependent way inhibits the ability to bronchodilate. If I would give an anti-IL-33, I could actually restore the bronchodilation capabilities. Now, you're all wondering, well, what about TSLP? TSLP doesn't have this capability. It generates airway hyperresponsiveness, but TSLP doesn't have the ability to inhibit bronchodilation. Why? The TSLP receptor is not expressed on airway smooth muscle in very large quantities, whereas ST2 receptor is expressed in high quantities in airway smooth muscle. So here's something that one of my colleagues in our institute has recently published 2 months ago. She's a rhinovirus C person, works with Jim Gurn. And so we get RB, we give RB to our lung slices, and just the infectivity of the epithelium, because it only infects airway smooth muscle, inhibits the ability to bronchodilate. So that's quite profound. This is a dose-dependent curve. And you can see the log EC50, less is better in the case of formoterol, but you can see when you infect with virus, and this is for 2.5 days, 3 days, you actually lose the ability to bronchodilate. So think about this. In my practice, I see people with viral-induced asthma exacerbations all the time. That aspect of the exacerbation is not very steroid sensitive. I mean, we treat with steroids, don't get me wrong, but I get bored and then I start to taper them because they're not getting any better and they're just, you know, they're angry at me because of depression and everything else. So I stop it, right? This could be a reason because you lose the ability of your bronchodilator to work. So you have sustained bronchoconstriction with no counteraction on bronchodilation.
Is that true of normal?
Yeah, that's true of normal also. So the corollary there, Lynn, I think is cough. You see post-viral tussive syndrome, and that can last for months. And so I think this is an interesting area of research. Now, TGF beta, going back to TGF beta, also has the same loss of the ability to bronchodilate. Here, this is diluent, you get beautiful relaxation. But in the case of TGF-beta also causes a profound decrease in the ability to bronchodilate. It blunts the ability to generate cyclic AMP. So if I measure cyclic AMP here with ISO, you can see in the presence of TGF-beta, cyclic AMP levels are profoundly diminished. Not only to the beta-2 adrenergic receptor, but also to the EP2 receptor, which PGE2 binds to. That's a whole different receptor, and it still causes the same inhibition of the response. So how might this work? Well, this one, Lynn, we have an answer. What we found is we did RNA-seq, and we characterized actually every type of phosphodiesterase found in airway smooth muscle. And what we found is in the presence of TGF-beta, there's a marked increase in a phosphodiesterase that breaks down actually the cyclic AMP. Here we block that response by using a TGF-beta antagonist. So it's really interesting that TGF-beta can induce a phosphodiesterase that then renders the muscle less responsive. This is fundamentally different than beta-2 tolerance. So what we have here is a complicated system. I'm sorry, you've made it through this far. You're almost there. This is putting it all together. So this is relaxation. This is how TGF-beta activates its systems. And this is a contractile agonist. What we know, and I didn't show you all the data, is TGF-beta activates SMAD. SMAD drives a phosphodiesterase expression of PDE4D, which is not really the isoform that rofumilast binds to. And that presence actually will break down cyclic AMP. So my argument has been beta agonists aren't the best bronchodilators. They're our only bronchodilator. We've characterized now new set of targets, the G alpha 12, which activates that pathway that causes contraction, Rho A and PI3K. Not going to show you all the data to support that, but I do have an interesting model here. This is human airway smooth muscle put into a 96 well plate, and we give a contractile agonist. Now, what we're trying to do is develop new drugs. We have the target, and this little Maltese cross, it's very cute, right? That's one cell contracting in a gel, allowing me to see relaxation. We take that, and smoke and mirrors, so it is early in the morning. We do that in 96 wells. We add a whole bunch, a library of molecules, and we ask the question, which one of those actually prevents the Maltese cross from occurring? And the one that we found, which is really interesting, is a PI3 kinase inhibitor called Cal-101. This is commercially available. It's a tool compound. But what you can see here, formoterol gives you bronchodilation. This compound can also relax almost to the same degree as formoterol. Now, why is that important? This target is unaffected by cytokines and unaffected by desensitization. A better bronchodilator. So here, all right, you don't have to believe me, Teal, but I'll show you. Here, I desensitize the receptor, okay? I lose the receptor. Now I give Cal-101. This was with salmeterol. Salmeterol is the best drug to desensitize the beta receptor to. It's wonderful. In the presence of Cal-101, look at that. And desensitization of the beta receptor. Beta receptor ain't working. This drug's working just fine. So then we go back to our slices. And we use Cal-101. Here's formoterol. And sure enough, although the dose response curve isn't quite as pretty as the one with formoterol, the maximum effect is quite effective. If we go down that pathway and go to Rho A, Rosin, or a ROCK inhibitor, has the same capability. So PI3 kinase, which is upstream, and ROCK, that is the target for here. We can get equal relaxation. So beta agonists ain't the best. We've come up with new targets. These targets are druggable. We can put them in inhalers to decrease side effects. We could actually prevent desensitization. So immunomodulation. I'm not going to go over this. This print is quite large, pun intended. These are all the things that airway smooth muscle can secrete. Whole ton of things, and I'm sure some of your favorite chemokines and cytokines are there. This is a paper Judy Black and I wrote. But I do want to give you this idea because it is important. And how do you generate steroid refractoriness? Here we gave airway smooth muscle, TNF, and interferon. And same thing here. And we're looking at fractalkine and CD38 expression. Now, unfortunately, hidden down here is if I gave a steroid, I could actually inhibit that signal. But when I add the two together, TNF and interferon, I can no longer inhibit this. So this is glucocorticoid insensitivity model generated by TNF and interferon. Here you can see it again. We're now giving steroids. This is with CD38 expression. You get activation with TNF or interferon. Here, in the presence of dexamethasone, this goes down. But when I add the two together, steroids don't work anymore. So interferon, TNF, at least in mesenchymal cells, can generate steroid insensitivity, not only to the type 1, but type 2 interferons also. So there's been a lot of work looking at the mast cell, smooth muscle cell interaction, the synapse. What we know is the airway smooth muscle changes the mast cell function, but the mast cells don't change the airway smooth muscle function. This is work done by Chris Breitling and colleagues, except for causing contraction. But it doesn't really change the phenotype of the cell. Others have shown, and we have shown, that T cells can actually modulate EC coupling directly. Don't have time to go over it. And they do have T cell antigen responses that promote migration and proliferation. So what I want to finish with here is, well, some remodeling. Again, airway smooth muscle grows. This is our central hypothesis. Given genetic predisposition in the environment, you get injury. That injury repairs in 85% of asthmatics, but because we think of an aberrant injury repair response, airway remodeling can become dysfunctional. That could also be epigenetic. This is a slide just showing the remodeling lesion. These are all the items that can cause growth of muscle. So GPCRs and RTKs, receptor tyrosine kinases. But let's now talk about new biomarkers. So where did I go? Talked about AHR. We talked about beta agonists. Muscle grows. Muscle can secrete things. It can become glucocorticoid insensitive. We're really excited about this. Over the last 2 years, we've been working on identifying the transporter. The ABCC1 transporter is a cassette that is a family of genes that are very important in cancer drug exclusion of the drug from the cytosol, but it's found in virtually every cell. Now, remember our friend, the beta-2 adrenergic receptor, we activate cyclase, ATP goes up, that activates PKA to decrease tone. What we discovered, and I'll show you the data, is a specific transporter in the airway smooth muscle actually excludes cyclic AMP. So we think, old school, is that all the cyclic AMP is broken down by phosphodiesterases into AMP and then into the component parts. I'm going to show you data that that's actually wrong. That this transporter degrades more or allows the egress of more cyclic AMP than phosphodiesterases can work. So here's the time course. I give for scolin, this activates adenyl cyclase directly. That's in the black here, and this is the total when I add it up for Motorol. In a time-dependent manner, these drugs increase cyclic AMP. Well, I made our postdoc do this experiment about 10 times because I said, how about we measure cyclic AMP in the extracellular space? And he looked at me and he said, well, you know, Dr. Panettieri, it's a second messenger. It stays in the cell. It doesn't get outside of the cell. I said, trust me, just do it, right? So we did it, okay? This is now the extracellular cyclic AMP over the sine time course. And look at the topography. Wow, it goes up. And it doesn't start to leak out of the cell until you get to 15, 30 minutes. At 24 hours, I have an enormous amount of cyclic AMP out of the cell. And this ain't getting broken down by phosphodiesterases. We believe it was the ABCC1 transporter that was responsible. The other exciting thing for me is the heterogeneity of the response. These are 5 different donors. And you can see basal and formoterol. First of all, there's a variety of responses to formoterol in an individual, heterogeneity of response. But you can also see the extracellular quantities vary substantially, especially at the basal level. Now, think about this donor too. If they're leaking a cyclic AMP, it ain't going to bronchodilate. So what we're looking at is the heterogeneity of the human response to leak cyclic AMP. This is with forskolin, and we see the same kind of variance in the baseline. So what if we knock it out? Well, this is now cyclic AMP intracellularly. I can use a pharmacologic modulator of ABCC1, and sure enough, the intracellular levels go up if I plug the hole, if I'm not leaking. What if I knock it out? Here, I get an enhancement. So what if now we do this and look at cell stiffness? Well, if I plug the hole pharmacologically or with siRNA to take out the transporter, look at this. The airway smooth muscle relaxes more because there's more intracellular cyclic AMP that activates PKA and relaxes the muscle. Same thing here when we're looking at cell stiffness. Those are the beads on the cell. This is with an ABCC4 and an ABCC1. ABCC4 is the second highest expressor transporter, and ABCC1 is the predominant. So if we do this in multiple donors and we look at knockout, you can see that there's a variance in one sensitivity, but by and large, everyone who has knocked out the transporter has less extracellular cyclic AMP, less inducible cyclic AMP, and the intracellular cyclic AMP goes up, but it's not uniform. There's differences in individuals. So with that, we were excited because nobody had reported that this transporter actually leaks cyclic AMP. And I had the crazy idea. I called Chef Chup. I said, hey, give me a bunch of your asthma samples in your well-phenotyped patients. Let's see if it's in the blood. Well, he gave me a bunch of these. These are from Yale. We did 16 asthmatics, 8 controls. This is sort of the characteristics and the severity. These were pretty affected individuals. FEV1 was 72%. They were mostly moderate, mild to moderate. They weren't severe. And lo and behold, in our publication, we found that the serum cyclic AMP levels were higher in the asthmatic patients than in age, gender, sex-matched non-asthma. But look at the heterogeneity of the response. Almost tracked beautifully with the airway smooth muscle cells that we had shown. Heterogeneity of response. Well, this excited me. You'll want to wait for the paperback. But, you know, really what I wanted to know is why. Some people have very high levels of cyclic AMP and why some have very low levels. Is that the heterogeneity of the response? So what I want to leave you with is a complicated model, but coming back to relaxation, I've shown you new targets to make better bronchodilators. But maybe if I plug a hole, the ABCC1, I could make beta agonists work better. Maybe I don't have to use a completely different beta agonist to improve outcomes. The good news is Airsupra, albuterol budesonide, was coming out at the same time. This is a combination rescue inhaler where you use budesonide and albuterol. Does that make sense? In this paper that we published in the New England Journal of Medicine, what you can find is there's less asthma attacks if one uses albuterol budesonide, SABA ICS, to improve bronchodilation. So this didn't exactly prove what we were talking about. It's heading in the right direction. So coming around to non-genomic and genomic effects, chronic is delayed, requires the GR alpha receptor to be activated, but there's a non-genomic effect of steroids that can occur acutely. We have a very sensitive assay to measure cyclic AMP, 1,000 times better than an ELISA. And what this does is it's a reporter that if this goes lower, you get more cyclic AMP. And here you can see formoterol. We can measure formoterol in the femta quantities and start to get an enhancement in the bronchodilator effect. Very nice. You can see it's a loss of signal, means bright, no cyclic AMP after agonist, the color goes down. Why do you reverse it? Because you have more sensitivity if you have an on-off rather than looking for an on signal. So what if I give budesonide? At the same time, I give formoterol. No way this is affecting gene expression. Look at the enhancement in the budesonide mediated response to formoterol. Budesonide acutely with formoterol is a better bronchodilator. Why would you ever use beta agonist by himself when this is bronchodilation? Now, that New England Journal of Medicine paper suggests, you're giving more steroid at a time when a patient's having a flare, so that way the anti-inflammatory effects are paired with, that's all right. But as a muscle biologist, it's all about dilation. So if I got a bigger, better bronchodilator in the presence of budesonide, I want to do that. Matter of fact, it works not only with albuterol, but anything that increases cyclic AMP, like PGE2 through the EP2 receptor. You see the same enhancement here. So this works regardless of the way you're increasing the cyclic AMP. Now, what about budesonide by itself? Well, we were shocked because budesonide by itself is not a great bronchodilator. It doesn't bronchodilate at all. But it does, using a sensitive assay, actually increase cyclic AMP by itself. So, and we believe there are territories in the plasma limit that allow cyclic AMP to manifest. Here,
is this true if people are already on high dose maintenance?
Yes, yeah, that New England Journal of Medicine paper, everybody was on standard of care on ICS LABA already. This is in addition to. It's all about timing. Here's that lung slice model. Budesonide at World Cup doses ain't a bronchodilator by itself. But formoterol is a great bronchodilator. So that was a lot quickly. We believe that there is a membrane mediated GR effect that doesn't require GR alpha. I didn't show you the data. We knocked out GR alpha and this effect is sustained. So it's either an orphan receptor or it's a lipid RAF stabilizer that allows the GS to be activated by beta agonists. I did a whirlwind tour at 7 a.m. on how cytokines can enhance EC coupling. We talked about EC coupling compared to asthma, non-asthma. Asthma intrinsically contracts more. It relaxes less. It can perpetrate and orchestrate airway inflammation. Growth factors can increase mass. We've discovered ABCC1 is a hole that leads cyclic AMP. If we plug the hole, maybe we can enhance beta agonist responsiveness. We've also identified targets that can be new bronchodilators that aren't susceptible to RBC, to desensitization, to IL-13 and IL-4. Ultimately, we think the non-genomic effects of steroids is going to change the topography on the way we're going to relax airway smooth muscle acutely by making a better bronchodilator. So, Len, it is always a pleasure to be here. A paradigm shift. This is relevant to any cell. Stimuluses that are pro-activation are always in the body countered by pathways that inhibit that. Homeostasis is what the body wants. In my case, it's airway hyperresponsiveness, and this is beta agonist sensitivity. This is a great team that I have that allows me to travel the world so that I don't threaten with me picking up a pipette man in the lab. So they said, please go travel, preach the word, but don't touch my experiment. Thank you so much. Right.
There's plenty of room for questions. Every word of it.
That made it a little clearer, right?
So the new biomarker that I misunderstand is it cyclic AMP?
Yeah, yes, blood cyclic AMP level.
How do you know it came from the lung? How do you know
Oh,
the
I
source?
don't know. I
Doesn't
just
matter,
measure
as
the
long as
blood.
it
It's,
correlates
yeah,
with
as
asthma.
long as it correlates with asthma. It's a beautiful bookend of my story, right? Because I show you the extracellular processes. It doesn't mean that the muscle's contributing to the cyclic AMP. It's probably a whole host of other things. But if your ABCC1 transporter is dysfunctional in trafficking whatever cells, this is going to be a problem. Cyclic AMP and PKA are thought to be anti-inflammatories in the right context, right? And if I don't have a lot of cyclic A because my ABCC1 is modulated, then I'm going to be a pro-inflammatory effect. So we've now, well, this week, I've completed 840 samples characterizing cyclic AMP in normal individuals. And then we're going to establish a normal range. Blood cyclic AMP levels are measured for hypoparathyroidism. I didn't know that until I started to do the literature. It's urinary, and then the urinary has to be correlated with the blood level. So there is an ELISA available. What I want to know is why we have heterogeneity. Why do some people leak and others don't? Could that be T2 inflammation? Could it be overuse of their beta agonists? All these people are on beta agonists, right? Is it a surprise that the cyclic A goes up? Well, it's a surprise that some don't have it up in the blood. Either they're non-adherent or they're uber bronchodilators and responsive. Yes.
Do viral infections change the density of these ABCC receptors?
No, we've looked at least to RBC. We haven't done all the viruses yet, but you know,
It's functional change, not a number.
No, it's not a numbers. These things are highly expressed. If you look at the PCR values, they're very, very high. So it's an activation state. We do know PKA will phosphorylate that transporter and make it leak out more. So in other words, if you increase cyclic A, the cell says, hey, cyclic A is high. I want it high all the time. So the PKA will phosphorylate specific residues on the transporter and make it more actively to lose. So it's complicated. Hopefully it'll give us another, 4 to 8 years of funding to figure it out. Very utilitarian response.
What is it about asthmatics that they're essentially born hyperresponsive?
We don't know. You know what? We're starting to embark on that. We're looking at the epigenetic marks now with Carol over because we have literally hundreds of fatal asthma cells and and she's going to look at epigenetic marks to see if there's an epigenetic signature that would suggest hyperresponsiveness or hyporesponsiveness for beta agonists. That's where we're starting. Yes.
Do you think if there's any reason to think some of the currently available biologics might be better for patients who have more of an issue with hyperresponsiveness, like Dupixent? Is that too simplistic to think that?
Yeah, no, Dupixent, because of the work we've done with 13 and 4, I would put that.
Sure, people
Yeah. Okay, yeah. So let me just reiterate. Would the biologics affect the airway hyperresponsiveness? Specifically with the data I showed, 13, and we usually use 13 and 4. It would make sense if I gave dupilumab and decreased 13 and 4. That would enhance the bronchodilatory effect, as well as maybe make people less responsive. Now, as I remember, I don't think they looked at AHR with methacholine, dupilumab. Now we know TSLP. They have looked at airway mannitol, which is a mast cell degranulator, and they've shown that TSLP can decrease mannitol-induced AHR. How that works, we don't know. But I do believe, and this is why I've been trying to push some of the companies to look at the bronchodilator effect in the presence of the monoclonal. My hypothesis, if I remove 13 and 4, hitting the IL-4 alpha receptor, I should decrease, I should improve my beta agonist responsiveness. So, that it would require a little more sophisticated study than just ad hoc look at their teeth, their phase 3 studies.
That's I don't know if you want me to comment about it. There's a lack of information on many of the biologics, but of course, anti-eosinophil effects have agents have no real effect on direct hyperresponsiveness, which is closer to what you're modeling with just looking at the smooth muscle. Then there was the Brian Lipworth paper, which is uncontrolled, and it has this pretty significant effect on indirect hyperresponsiveness, which maybe occurs in the absence of some of the changes that you're describing.
Yeah.
But yeah, I think, unfortunately, in the literature about biologics, they, with anti-TSLP, they looked at airway hyperresponsiveness more than was the case with some of the other agents and so forth. And I think that going back with, I realize that there are reps from different companies here, but my understanding that, obviously the anti-IL-13 effects that you're showing in the dish there, are probably relevant to things like Dixon, but it wasn't studied that extensively.
Right. Yeah, I'm a big fan is try to understand in the human whether you can restore beta agonist responsiveness or enhance it in the presence of these biologics. I mean, people don't think of it that way, but it makes a big difference to a patient whose beta agonist wasn't working so well in the presence of T2 inflammation, and now you remove it and they suddenly respond better.
Yeah. But I was also, I think I mentioned this at the Aspen Lung Conference, that the 33 effects and 13 effects and how they interact with each other. You said that the 33 was not through IL-13.
No. Matter of fact, we did those experiments. We gave IL-33 and then took out, the IL-13 effects using a monoclonal antibody or a blocker. And we still got the IL-33 effects without IL-4 and IL-13.
Yeah.
So
that's really, obviously interesting for our lab, because we've shown how the epithelium regulates 33 and then through mast cells regulates type 2 cytokines. And so you would expect there to be a kind of back and forth, a negative and a positive regulatory loop, which is maybe why the New England Journal paper blocking 33 and blocking IL-4 13 receptor actually had
a deleterious effect. I always asked Mike Wexler, and he said, "Well, you tell me your impressions." I said, "Well, my impressions. Why'd you do that experiment?" He said it wasn't up to me. So it never.
Trying to point out that we have a question in the chat.
No, okay. Let's see if I can do this. All right. Many with persistent asthma are either on baseline inhaled steroids or added increase with yellow zone. How would albuterol budesonide be different? Thanks. Yeah, that's a great question. So all the Air Supra studies were done on standard of care. Some were on low dose, some on moderate, some on high dose ICS LABA. But, again, I think this is all about timing and dosing. So the non-genomic effects of steroids needs a fairly hefty dose of the steroid given simultaneously with albuterol. So my simplistic thought is the baseline ICS LABA does a great job at some control. But when that control is overwhelmed, you need to give the budesonide at a heftier dose simultaneously with the albuterol to get an enhanced bronchodilator effect. So that would be my answer there. Great question, though. Yeah, there is one more, but my fingers are too big for this. Let's see. There. Why is my name stuck on the screen? We have some very accomplished individuals here who can answer that question. That's not in my wheelhouse. We like you, Jen W. Any other questions?
All right, go. Thank you.
Thank you so much.
Now, as many as possible stay. We're going to present a clinical case to follow. Those who are online who want to stay, I presume we can broadcast the presentation as well, but I'm encouraging everybody in the room to stay here, to stay if you can, if you don't have a clinical responsibility. Jocelyn, you're going to present the case for us.
Gonna grab some coffee and
Okay,
listen. to
everybody stand for a minute and. We've got 12 people hanging in here, I guess.
They were saying 12 people are staying on for the can I get access to my email? Absolutely. So what you do here,
One of the things, just before you do too much stuff, is there a way as he's presenting to get rid of this lower bar?
yeah, you just when you share a screen and you come up here, let's say, you go this part and then shrink down.
You just need to click the screen maybe and like it'll disappear. Or no, because I mean, because you were, I don't know, you were outside, so you didn't notice it. But there was a number of slides where basically this portion of it covered the bottom data, and so like if you were looking at it like logging in remotely,
Yeah.
you could see the whole PowerPoint, but on this screen you could not. So you understand what I'm saying?
I do.
Okay.
Well, let's get her going real quick and then we'll figure that out.
Yeah, I just wanted to mention.
Yeah, for sure. Okay. You don't have is it, what email did you need to use? Is it Outlook? Outlook. Is there a way to get it online?
Yeah. I'm not sharing my screen right now. Yeah.
So you go up to Zoom, just put stop sharing. May I real quick?
Yes.
Everybody's got your passwords. Okay, so this one is just sharing. Get rid of that. Oh, I put it that way and she'll go away. There you go. Is this on presentation? It's okay.
hopefully you all are seeing presentation mode on Zoom. All right. My name is Jocelyn Cooper. I'm one of the first year allergy immunology fellows. I'm presenting a complex asthma case that I saw with Dr. Coker here.
Louder, I don't hear much.
Oh, sure. Presenting a case on asthma. So we saw a 40-year-old female with history of asthma, allergic rhinitis, eczema, and GERD. She presented to our allergy clinic after a few years of being followed by an outside pulmonologist. She had mild asthma in her childhood, but it progressed in about 2018 to a severe persistent asthma picture in her adulthood, which has been difficult to control. She's had several courses of oral steroids each year, multiple ER visits, and 3 hospitalizations for exacerbations during this 5-year period. She's never been intubated, but her triggers seem to be upper respiratory infections, exercise, environmental allergies, and overall she reported that these symptoms got worse when she moved to the Midwest in 2018, and overall tend to be better in this area of Seattle. In terms of her treatment for allergic rhinitis, allergy immunotherapy was a thought for her. She's sensitized to trees, grass, weeds, and dust mites, but her poor asthma control had been a limiting factor to getting this therapy. So she's taking an oral antihistamine and a steroid nasal spray every day. She had extensive workup for really other causes of her symptoms of asthma, which from what I gathered were shortness of breath and some sort of nighttime symptoms that got better with steroids. She has GERD, and that was treated with a surgery anti-reflux procedure, and she's now asymptomatic from that perspective. She had an evaluation by ENT for vocal cord dysfunction, which did not show that. She also had an extensive cardiac evaluation and did not have pulmonary hypertension on her workup. She's never smoked. So the earliest PFTs I could find were from a note from a pulmonary doc. Unfortunately, there's no bronchodilator challenge, but essentially what you can see is that her FEV1 over FVC is normal, and the FEV1 is over 100% based on the values of her similar patient profile. So the year after that, she had a methacholine challenge done, and from my understanding, she was on her current therapy, which was Dupixent, an ICS LABA, and Pulmicort. I couldn't find the reason why, but I imagine that with her severe uncontrolled asthma, she was not able to go off of her treatment in order to do this study. It was negative, but overall she carried this clinical diagnosis of asthma based on her response to corticosteroids.
I don't know, but this seems pretty confusing to me.
Well, it's very confusing to me. I mean, so what is the evidence that she's got asthma? So,
Sure.
I mean, everything you just painted a picture that doesn't sound like asthma, but FEV1 FVC is extraordinarily well preserved. One negative methacholine is pretty damning for not asthma. A second one would put you into about 98% likely not asthma. But she responds to steroids, right? Did we get the physical exam? Maybe I missed the BMI. Was she big woman?
Not particularly. I think she does have sleep apnea, but no, not particularly. But
no, I mean, have sleep apnea. I mean, usually you're moderate with obese. BMI was in the 30s.
I'm not sure. Below 30.
Yeah. So not World Cup, but substantially fast going down.
So this was her workup initially, kind of to evaluate her phenotype. So she had a not a very impressive absolute eosinophil count. Her total IgE was 3 at the time of our meeting her. It was highest in 2019 at 22, and her pheno was never very high either, so it was 9 when we saw her.
Sounds like the perfect patient for now.
Is that pheno while she's on inhaled steroid?
I believe so. I think a lot of her diagnostics were done while she was on treatment. I couldn't find any evidence that she had stopped treatment for these studies.
But that's a very important point. You know, you could say you take off our medicine and see what she has, but let's say that was correct. She's on medicine and still has symptoms, right? Still gets to the issue of whether it's asthma, but you know, something appears to be uncontrolled, whatever the disease is.
Right. She also had a high-res CT done in 2019, which interestingly showed some sort of mosaic pattern that could be consistent with air trapping and chronic small airway disease, but she didn't have any fibrotic changes of note. Her therapies were Symbicort, BID, and Spiriva 2 puffs daily, which weren't quite controlling her symptoms. She had also been put on azithromycin 3 times weekly by her pulmonologist for what they were considering like a T2 low phenotype, with her low pheno, low IgE, and lack of eosinophilia. She was also interestingly put on dupilumab in 2019, and it seemed like she was on that for several years with some improvement, but not really full. She does have eczema, but I believe it was put on for her asthma.
She's been seen in multiple tertiary care centers. So she has worked in many different states, and there seemed to be some worsening of her symptoms based on her geographic location. She does have environmental allergies as well. And so the pulmonologist initially got it approved on the diagnosis of corticosteroid-dependent asthma, because she, after having so many hospitalizations and tapers, was on daily prednisone for a time period as well.
She was OCS. And what was her skin prick? Positive to
Dust mite, trees, grass.
She was on antihistamine, so I confirmed it on serum IgE testing.
Positive. Got a total IgE.
Very low.
Yeah, it was really low, right. But there's a perfect example of sort of the uncoupling on the skin prick from the total IgE.
For those on Zoom, if you didn't hear that, we were just talking about her concurrent allergic rhinitis and the reasons why she was put on dupilumab.
I'm sorry, she had an ORL evaluation looking for polyps, ORL, ENT.
I don't know, actually. Do you know?
There's no history of nasal polyps. She's been evaluated for that.
She has, okay, great.
Oh, okay, thanks, Delara. So just to touch on the methacholine challenge, and feel free to chime in, but just it's the PC20 is what we measure provocative agent concentration to drop the FEV1 by 20% and less than or equal to 8 milligrams per ml is consistent with asthma. Her test was negative, notably, but in a few studies, it's been found that the sensitivity of that test can be less in patients who are being treated with controller medications such as ICS and LABA. It could also be normal in patients who only are hyperreactive in the presence of their triggers. And there are some lower sensitivity in white versus African American patients, some higher sensitivity in atopic versus non-atopic patients. Those
The sentence there can be normal without recent exposure to triggers. I don't like Ray's opinion on that after just telling us that asthmatics are hyper-responsive from birth.
Yeah,
It seems
well,
very strange to me.
Well, the statement is true.
Yeah.
But you don't take somebody who's, gets a trigger, who becomes wildly methylcholine sensitive. You're in that gray zone, and it might move maybe 2 logs or 1 log, but it's not somebody's going to suddenly be, in the highest sensitivity category because of a trigger. I mean, viruses can give you a false positive intermediate methylcholine challenge, But I would have thought this woman would have been high, like a severe asthma, given all the medicine she's on. One other important question is when measured the FeNO, was that on dupilumab? Because FeNO will decrease the, dupilumab will decrease your FeNO. So maybe her FeNO was 40, I don't know. Historically,
I think there were multiple phenos measured. They were never greater than 10 or 12, I think. And she was on dupilumab for several years during that time that she was getting diagnosis, or further studies. So again, her PFTs in 2022, when we met her, appeared normal. Her DLCO was also normal. No evidence of restriction either. In our clinic, she was started on tezepelumab 2 months ago, and
so
she's
before you get
oh,
the
sure.
therapies, so let's stop there and figure out what she has. Because there's six biologics you can choose. So why? So did you feel she had asthma?
I think maybe it was she came from an institution that has a very strong pulmonary program, and maybe that's a bias that I felt like, okay, she probably I know. She probably I felt like, I believed it that she had asthma, and talking to her about her symptoms, having wheezing, getting steroids and feeling better, kind of all fits with asthma, but it is confusing, her studies.
Well, I think, the CT scan, we didn't see the CT itself, but, the mosaic pattern, things like that, you start to think of bronchiolitis obliterative, things that would cause shortness of breath. That's different than asthma. That's what I thought of immediately that this woman had food. But then the DCO is normal and really have much restriction. So those would be my differential diagnosis. You ruled out VCD, obviously something very high in the differential. But everything you showed me, I'm unconvinced qualifies as severe uncontrolled asthma.
Yeah, I think when I saw this patient, I had the same question. And for me, she had such an extensive evaluation, having some in front of the patient for sure, had ruled out VCD. And when I talked to her and ignored the objective data, to me, it was like, this sounds like asthma. Okay. You have asthma triggers, worsening of viral infection, response to oral corticosteroids, and she had paper inhalers, she would symptomatically get worse. When her kids would get sick, she'd catch it and then have a flare, wheezing, cough, shortness of breath. Although objective data, argues against asthma being the diagnosis, when I saw her and talked to her, like clinically, to me, it sounded like, this sounds like asthma, but I have the objective data to support it.
Right. And she's a dupilumab, yeah.
Dupilumab failure, yeah.
And I know you're talking about some of the other biologics, but given her phenotype, a lot of those like mepolizumab and other things were not tried for her. So that's why we went for tezepelumab a couple of months ago, and she has had some improvement in her symptoms. Thus far, she's been able to stay away from the ER and oral steroids, and she was able to stop her Spiriva. We continued on her Symbicort. We haven't tried tapering that because we're doing we're starting Odactra, so sublingual immunotherapy for dust mite allergy. Ideally, she could be on allergy shots, but she's has a busy schedule. It's not very feasible for her. So this is kind of what we're trying. We're trying to optimize her asthma treatment while treating her concurrent allergic rhinitis.
Getting the Odactra approved.
It was approved.
Yeah, we gave her first dose in clinic. She started well. She's on home therapy.
Good. Hard to get it paid for. I don't know how effective it is. The true feeling is, I think, oral immunotherapy works in glass. It's not very good for dust mite.
I think it's better than nothing. For this patient.
But it depends on the value proposition that managed care company. Better than nothing.
And then this is just her most recent set of PFTs. I believe this was done before tezepelumab started.
This is a tough case in that we like to show objective evidence of improvement, right? So OCS burden, well, that's pretty easy. You can count the numbers of milligrams that the patient's on over last year, numbers of exacerbations, both hospital, ER, and urgent care. That's pretty easy. The FEV1 is not going to be very sensitive here because she's got great pulmonary function. I don't know about her ACT score. I guess whatever score you're using is probably a learning curve because she's got lots of symptoms and not a lot of objective evidence of disease.
In these types of patients, I try to ground it in like the things they can do now that they couldn't do before, kind of going back to the success thing you mentioned yesterday. She's able to play with her kids more frequently, go outside, and started exercising. So there is a dramatic improvement in her tolerance to do these different activities that are meaningful to her. And then I would count no ER visits and oral steroids as a win because, you know, as we know, like going to the hospital system and getting put on steroids is not a benign thing. So.
No, and it really exposes them to really, you know, a whole host of complications. So now you've decided to pull the trigger on Tezspire, right? And you killed the Dupilumab. So how do you know the drug is working? You can't ask the patient. And no, you can't ask the patient. No, you don't want to confuse the issue with that. No, you can't ask the patient, but how do you know? And the reason I'm asking you is because you weren't at my talk yesterday. I use a term that describes engagement of the target, like a pharmacodynamic target engagement. So what would you look to from a laboratory standpoint to know the drug works?
I'm just gonna maybe move on to. Well,
Another fellow.
Yeah.
I use the name, so I. The last 30 seconds, if you get up to in the last 30 seconds, I'm happy to weigh in.
The way we do this, when I had my lab meeting or whatever, and the presenter always has a coupon, and that coupon can be ask a friend, and the friend has to be at the same level. And then if they don't get it right, they can ask the next level up. So we have graduate students talking to postdocs, to junior faculty, and then it's amazing how the junior faculty walk out. Well, let's think about it, right? Tezspire, you know, Tezepilumab can modulate, you know, really 3 pathways, right? It's going to modulate the eosinophil count. It's going to modulate the pheno, and then it can modulate total IgE, actually. In this case, total IgE is negligible, but I'd be very curious, could you make her, you know, non-atopic or non-sensitive using specific Ig? That hasn't been examined in the case of Pezzy, but it would be really cool to see if that's the case. So that would be, so if she had lots of EOs and pheno, you gave her Tezspire, bring her back in, the pheno goes down, the EOs go down. That means I've engaged the target. Doesn't mean I improve the patient, but I know the drugs work.
right.
Then you ask the question, if they didn't improve, then these things were true and unrelated to the patient's disease process. If they are, then that's hallelujah, throw your inhaler away and walk, right? I mean, that means you've engaged the target, the patient's better drug. Same thing with IL-5, right? If the IL-5, when the EOs go down, they don't go down, that's a problem. Other patients, Lynn, I've seen, and I'd love to pick your brain. I get people with World Cup phenotypes, 80, 90, give them steroids and the pheno doesn't. And I know they're taking it because they're having side effects from the steroids. I don't understand that at all. When I talk to the pheno mavens, they just said, yeah, we see that. I said, that doesn't help me understand.
I haven't seen.
Yeah. So maybe I don't know why that's the case. And
You talking systemic steroids or inhaled?
no, oral.
Oral.
Yeah, not I'm talking oral steroids. And it should drop like that.
Yeah, like a rock.
Like a rock. It doesn't. And so, sorry, I'm stealing your fun. Go ahead.
Oh, that's okay. This was the last slide. I just wanted to touch on the phenotypes that we were thinking about in her T2 high, T2 low, and how it might guide your choice of medication. But because she's not in the T2 high category based on her labs, theoretically these dupilumab and mepolizumab wouldn't necessarily be good options for her. But with the T2 low and a different set of cytokines going up higher on the pathway, tezepelumab seemed reasonable. And so you all are probably familiar with tezepelumab, but anti-TSLP intended as an add-on therapy to treat any of these asthma phenotypes, and then should, based on the study, reduce asthma exacerbations. And so we kind of touched on this. This is a very general pathway, but just showing that it is upstream of the eosinophilic or neutrophilic or allergic inflammation that she may have contributing to her disease. And that's it.
Well done.
Thanks for listening.
Yeah, I would argue though. I would argue that she's not an exclusive low T2, right? Because she's allergic to a bunch of things, so that would put her in the atopic T2 inflammatory group, not a low T2 exclusive. So I mean tezepelumab's a great drug for that because you bracket both things.
I think the interesting thing is we think of Tezspire as T2 low, but I mean, if you look at the trial data, I mean, people with T2 high respond well as well.
Well, they respond better, frankly. So, you know, this is a T2 high, T2 low for us. I don't say it's exclusively T2 low, mostly because we don't have anything there, and this is the first study that showed it worked in that area, so we think of it as a low T2, but I think of it as a T2 high, T2 low. So maybe she got, you know, some of her Th17 cells affected as well as some of the other pathways.
And I think the other thing that this case raised for me, and when you look at the total Ig, you think of localized Ig production in the airways, upper airway and lower airway, and then of course what's bound to the mast cell in the skin. So a low serum Ig doesn't always necessarily correlate with what's happening in the airway or in other parts of the body. And so I think that if you go based off the IgE, you might think she's not very confident when you skin test her and blood test her and she has positive testing. Yeah. And so I think that was another point as well.
I think that's a really specific
Greater than 0.35, so you know you meet the threshold for positivity, but they were positive to grass, the types of grass, Timothy, Bermuda, and dust mites as well. Mold testing was negative because in a severe asthmatic, I was asked about ABPA, and it's my bias as an allergist. So,
no, it makes perfectly good sense. I would have thought, you know, I mean, you did the requisite work with the CT scan too to see if this is stuff like that. So I thought that was great. You know, it's one of the things I preach to our pulmonary fellows because you know they always look for a high IgE and they say they're atopic. I said, and then they do the RAS test. I said, you know, you can't say that. You know, you get led down the garden path because the way to start on lizumab by having your IgE and BMI to dose it. That doesn't mean atopic. So.
Yeah, I guess I'd ask your opinion from pulmonary side on asthma COPD overlap, which is its own, I think, like 10-hour talk.
No, not really. I mean, 30% of asthmatics smoke. So you're going to get, we tend to see a lot more of those patients than you, right? If somebody smokes and they come to you with uncontrolled airway disease, you usually send us, which is fine. But, you know, and then there's a fair number of people who are going to be allergic and have COPD predominantly. So, you know, this overlap is somewhat confusing. So I didn't see that she was a smoker.
She's not, never, yeah.
Right. We have pulmonary fellows rotate with us, and I guess what I try to impart them is to always think about allergies and asthma, even the COPD patient, because there are no biologics approved for COPD.
Not yet. But no, I would agree entirely. I think these are different diseases. You know, we think that it's a continuum. There is a continuum in asthma from reversible asthma to irreversible asthma. That isn't COPD. If you're a non-smoker and you're not biomass burning anything, you can't get COPD. I mean, in the US, that's not COPD. That's irreversible asthma. That might have been from all the things, Len, you mentioned, the airway remodeling that's gone on for years that's been uncontrolled. That's not COPD. COPD in US is driven by asthma or alpha-1 antitrypsin deficiency, ZZ or MC. You know, I view these as separate diseases But any human can have as many diseases as they want, so they can be allergic. 30% of the US are allergic. And then you're gonna have a smoker who develops airways disease. And so, we do know, I think the biggest innovation is to use inhaled steroids only in the exacerbating COPD patient rather than everyone, because everybody was on the purple bagel before. Everybody would get all 3 doses. It's good for you. Just take it. But what we know is that we're doing a lot of harm with because of causing pneumonia and COPD. So now the focus is only on exacerbating. The dupilumab study is really exciting. The first positive study in COPD. So they have to do a second phase 3 study to see if it's going to get approved, but it was a robust signal. However, that's the good news. If you looked at the details, who did they enroll? People with reversibility and high EOs or T2 inflammation. Well, like I said, anybody can have 2 diseases. So more to be told. The COPD piece, just so you know, we talked about phosphodiesterase. There's going to be an inhaled phosphodiesterase inhibitor, PDE3 and 4 inhibitor, in an inhaler, which is very exciting, and it doesn't have the untoward effects of Rafimilast, which is GI symptoms. So that'll be coming out in COPD, not in asthma. But because PDE3 is found in airways and muscle, 4 can get induced with TGF Beta, you think it could serve as a bronchodilator also. It'd be interesting.
I think the proof here is if you continue to reduce your other meds, what if you don't therapy?
She came off Spiriva already after starting Tespire. She tried to wean this in before, but then got a cold, and so we told her not to. And I might try again when I see her follow up.
Yeah, I think getting her off medicine is helpful for total body exposure to systemic steroids to help her long run. To exercise also keep her bone mass up. That's a great case.
Thanks so much.
you
Appreciate
for
it.
presenting. All right, thank you for